Hi everyone, thanks for checking out our video. In this episode, we'll be sharing the experience of using optical tweezers. So, what are optical tweezers? Are they light beams to pluck your eyebrows? Of course not, silly, but you're quite close. As you can see in this simulation, highly focused laser beams are illuminated onto the microparticles to either move them or trap them. The laser beams generate two forces. First, the scattering force. This scattering force pushes the particle along the direction of the beam propagation. And two, the gradient force. This gradient force attracts the particles towards the focal point of the laser beam. So, what equipment do we need? First, a high-tech video monitor, a control box, a laser, a fancy microscope, a sample, and a CCD camera.
Here, we will tell you a little about the equipment we used in our experiments. We used a helium neon laser for which we adjusted the voltage of the beam. The laser beam bounced off several mirrors and then entered the beam slitter before reaching the sample. In our experiment, we used an inverted light microscope. The light source was positioned above our sample, while the camera for image processing was below the sample stage. The images recorded by the camera were played on the TV monitor. The sample we used contained styrofoam spheres suspended in aqueous solution. We moved the sample manually using the stage for the microscope. In our first experiment, it was our task to make a shape or drawing using the optical tweezers. We attracted particles to the laser beam and then moved the stage gradually to move the styrofoam particles as we desired. By moving the stage too quickly, the particles would dislodge from the laser beam. So it was important to move the stage slowly to keep the particles within the laser beam. Once the particles were released, which was done by moving the stage quickly, the particles continued to vibrate and some would drift out of place. This made it very difficult to make any recognizable shapes, but we did. The blue square is drawn on the TV monitor so that optical microscope users know where the laser beam is focused. There, we can see a single particle that is trapped within the beam of the laser. Can you guess what our shape is? We created a Big Dipper. Next, we had to determine how changing the voltage of the laser would change the number of particles that were attracted to the laser beam. As we increased the voltage, more particles were attracted to the beam, as can be seen in the above graph. After a certain voltage, too many particles were attracted to the beam, and we could no longer count the number of particles that were there. A metallic microsphere has a large number of surface electrons. These free electrons can absorb and emit light very quickly. Therefore, when the laser is directed onto the sphere, the light scatters in many different directions and cannot control the movement of the sphere.
The following diagram shows a focused laser beam incident upon a spherical air bubble in water. So which of the arrows indicate the direction of the net force acting on the bubble? Since the refractive index for water is greater than that of air, light beams entering the air bubble will deviate away from the normal. Since both beams are going in a general downward direction, the net force will act downwards too. The answer is C, a downward resultant force on the air bubble. When light moves from a medium with smaller refractive index to a higher refractive index, the light moves slower, so the beam bends towards the normal. Therefore, we will have to draw the normal perpendicular to the surface. In other words, going from the surface to the center of the sphere, in one color, and the beams for A and B in another color. The sphere is pushed to the left, F net points left, because there is always a restorative force back to the center, which also means the sphere is pushed towards the center of the lens. Due to the precision of optical tweezers, users are able to study molecules or atoms at much smaller scales than before. This opens up the door for the study of DNA, cytoskeletons, and measurement of femtonewton forces, just to name a few. One key idea behind the techniques to achieve such precision is a special light modulator. To visualize this, imagine a cyclist being trapped on the spot by winds blowing from all directions. Well, that is what we'll be achieving with the optical tweezers. In the fields of biology, optical tweezers contribute greatly in the studies of cell sorting, cytoskeleton probing, and DNA manipulation. Cell sorting is when a population of cells are divided into smaller groups based on internal or external target molecules. This process has a very high recovery rate, which is very important for future physiological applications. The target cells can be moved by optical tweezers in a non-invasive manner. This design is based on dynamic fluid patterns and dynamic light patterns in which single and multiple layer traps are employed for cell transportation and recognition. The optical tweezers technology is also used with the actin cytoskeleton model to probe the distinct underlying remodeling mechanism. This allows scientists to quantitatively study how cell mechanical behavior is associated with alterations in the cell functions. The translocation, or rearrangement, of DNA molecules is an integral process for the functioning of living cells. Many of the basic physical, chemical, and biological interactions have not yet been studied because they are not directly experimentally accessible. Scientists have shown that the combination of optical tweezers, single solid-state nanopores, and electrophysiological ionic current detection enable deeper insight into the behavior of DNA molecules. In the fields of biophysics, optical tweezers play a part in femtonewton force measurement and atom trapping. Several generations of technological improvements and refinements have brought optical tweezers to their present-day state, a cornerstone technique in the life sciences, employed for both micro-manipulation and the accurate tracking of piconewton forces generated in biology at the subcellular level. The manipulation and force sensing using optical tweezers is contact-free and sterile, since no foreign objects are in touch with the often delicate sample. Last but not least, atom trapping. Single atoms are promising candidates for physically implementing quantum beats, the fundamental unit of quantum information. Optical tweezers have played a key role in cooling, trapping, and imaging single atoms.